Okay, well, thank you for that introduction. The title of my talk today is From Community College to a Biophysics PhD, A Path Less Traveled. So, as Emily mentioned, I am a structural biologist, so I really like it when things are organized in a really nice way. And so to go into that, I'm going to tell you about the organization of my talk here, because if there's any sort of disorder whatsoever, I get very nervous. So I'm going to begin my presentation sort of talking about my biography, about like my path through education to get to this point where I am now. And then from that, we're going to kind of take a sidestep into talking about the method that I use for my research, which is cryo-electron microscopy. I'm going to give you kind of the long and short of how it works and like why it's cool. And then from there, I'm also going to give you a brief introduction about a class of enzymes that I work on, as was briefly mentioned in the introduction. And then finally, we're going to dive into the science, a little bit about my research and a story that I just recently completed. So I started education at the City College of San Francisco. CCSF is a community college. I actually took a few years off between high school and undergrad. I wasn't really sure what I wanted to do right after high school, and so I went to work basically, and then just tried to figure it out. I'm glad I didn't do that, because if I had gone straight in with my original idea, I'd probably be a lawyer at this moment, which as Emily kind of talked about, probably wouldn't have been the best thing for humanity and the world, that kind of thing. But I eventually decided that I wanted to study science, and that I was really interested in chemistry, and that I was going to go to community college. Not necessarily, because that's just like the most common way of doing it, but basically I didn't want to start at a four-year school because I was kind of concerned about how much debt I could rack up if I went straight in, and if I decided to change direction at some point, well, I might end up spending more time than I needed to and spend a little more money on tuition. So this seemed to be a really smart way of doing it. I was able to actually get all of my tuition paid for through this program called the Board of Governors Fee Waiver. Now it's called something different. It's the California College Promise Grant. So if there's any community college students in here who are paying credits, you should look at this. It's pretty easy to get, and they pay for everything, which is really nice. So like I said, I was taking classes in chemistry, but I was also part of this program that was focused on biotechnology. It was like an associate's degree sort of program. It was a bunch of classes that were laboratory-based. It's the biotech program. And so, you take classes about mammalian cell culture, fluorescence microscopy, stem cell science, that kind of thing. And the idea is that it prepares you for a career to become like kind of a lab technician at some of the other startups and large biotech companies that are, prolific in the Bay Area. But for me, this was a really good opportunity to sort of learn some scientific techniques a little bit ahead of time, kind of put in practice of the theory that I was learning from my regular undergraduate courses. And this actually had some benefits to it. So I joined a group that was focused on genetic engineering and synthetic biology sort of paradigm. And this was a competitive sort of organization that allowed us, the first community college team, to actually go and compete at this kind of competition at MIT. And it got us a little bit of attention from the New York Times, which was pretty cool at this point in my career to actually get some sort of like feedback from the public about that. And so this was a nice thing that arose from that. And then from this point, I transferred to the actual four-year school that I'd been putting off at UC Santa Cruz. And the way that I got here was through a Transfer Admissions Guarantee Program, or TAG program, which is another great thing that you guys should take a look at when those of you who are considering transferring to a four-year school that haven't done so already. This program guarantees you admission to one of 6 UC campuses from a California community college school. And as long as you maintain a certain GPA and you meet these prerequisite course requirements, you're going to get in, and you can pretty much have that, on the back burner at that point. So I used that to get into Santa Cruz, and I continued working towards my degree in chemistry. And the second semester that I was there, I realized that I really need to get some kind of research experience, because I was interested in graduate school at that point. I'd figured out that I really liked science. I didn't want to leave it anytime soon. And so this was a really big step into getting into graduate school. As Emily pointed out, and we talked a little bit about earlier, you got to get that research experience. So this was my first mentor. This is Carrie Parch at UCSC. She was teaching a second quarter biochemistry course on kinetics and structural biology, and I sort of just kind of fell in love with the science that we were talking about. And I found out that she works on circadian proteins, which are basically the proteins that are responsible for the cell being able to tell time. It regulates a lot of areas of biology, and she's interested in sort of the physics about how all these proteins kind of come together and transmit that signal. And so I asked if I could join the lab, and she said yes, which was a really great thing for my career. So early on, I was able to get some pretty good research experience focusing on cryptochromes and this protein here called period. I was able to write up a pretty decent undergraduate thesis, which was a requirement for my degree, and we also got a pretty nice paper out of that as well. And then I was also able to sort of talk about it at an international conference that was pretty close in UC San Diego, which was also quite convenient. Now, it wasn't just really the research experience that was important about this kind of point in my life. Carrie was a fantastic mentor and actually still is. We still correspond quite readily, and we actually collaborate now too. But this was a person who cared about who I was and about what my sort of trajectory was going to be into the future. And we would have long talks about sort of the steps that I needed to take, not just as an undergraduate, but years and years into the future, about if I wanted to be successful as a research scientist. And so I really attribute a lot of my success to kind of this early mentorship. And I would really emphasize that it's important to find someone like that if you want to be successful in science, someone that cares about your trajectory. Now, from their mentorship, she told me about this really excellent graduate program called Scripps at the time. And so I'd heard a little bit about it, and I asked her more questions about it. It seems like, wow, they have some really amazing research going on. It seems like right up my alley. What do you think? And she's like, yeah, you should totally go for it. And so I applied to also a lot of graduate programs, probably way too many. I didn't think I was going to get into Scripps. It was more of my reach school. And then as soon as I got the admissions letter, it was pretty much, well, that's where I'm going. But I still went on all the interviews too. I actually enjoyed the interviews. They were pretty nice. You get wined and dined, and everyone asks you what you're doing. So and you get to fly all over the place. So it's pretty fun. It is tiring if you do a whole bunch of them in a row. So yeah, I gotta have stamina. But while I was there, I did a bunch of rotations, and one of the rotations was with Gabe Lander, who is pictured here, and he is known within the community for being an expert on cryo-electron microscopy. And I had a pretty good rotation there, and I decided I was going to stick around and actually joined his lab. And so as we go into the next sort of section of my talk, all of the slides that I'm going to be showing you about how electron microscopy works is actually taken directly from his PhD defense. So he made all these movies. I didn't do them, unfortunately, so I can't claim credit. But I am going to explain it in a really good way. So electron microscopy involves these instruments that's here. This is actually the microscope that I use most often for my research, Talos Arctica. And it's a structural biology technique that really came from a material sciences background. Electron microscopy has been around since the 1920s, when it was mostly used of looking at materials and really small things, but more of a crystalline sort of nature. Nowadays, one of the big sort of things that you hear about in the press is how it's being used on biological molecules and biological samples. And it was recently recognized in 2017, I don't know if I can see that, by the Nobel Committee for being awarded the Nobel Prize in Chemistry because of how it's pretty much revolutionized biology, and specifically molecular biology. And so here's how it works. So first off, we start with a test tube, or an Eppendorf tube in this case, and within the Eppendorf, we will hopefully have a homogeneous sample of one or at most, a couple of different protein species. This is actually cryo-EM in this case. And so they should be at high concentration. They're moving around. I mean, they have kinetic energy just from room temp at that point. And what you want to do is take this sample and you want to actually vitrify or freeze in a non-crystalline manner these proteins in sort of a monolayer, where they're randomly distributed kind of in this nice homogeneous fashion. And once you have that sample that's ready, you irradiate it with an electron source. And so these green lines are basically just the electrons that are coming in, interacting with the protein of interest, in this case cryo-EM. And what you see here is actually the electron micrograph that's below. So hopefully your protein is going to be randomly oriented within your sample. So what I mean by that is it's going to be at these different sort of angles. So you can see here we have a nice cylindrical thing. It's a little bit tilted. This is a side view, for instance. And when you hit it with the electron beam, it's going to give you these sort of different images. They're not really images necessarily, because this is a transmission electron microscopy. So actually the electrons are interacting with the inside of the protein as well as the surface. So you get some information about the interior as well, which is really important for the type of stuff that we're interested in. Now, once you have a micrograph with all these different orientations, what you can do is cut out the images of each little protein, and then you can actually align them by giving it a certain orientation angle, and then you average them together. And when you average them, you actually increase signal in this case. So everything that's similar between them is going to be amplified, and you're going to have a signal increase, and the differences are actually going to be averaged away. And this is really important because carbon and water end up having a very, very similar sort of interaction paradigm with electrons, and so our signal-noise ratio is very low in EM, and so anything that we can do in the image processing side is really important because we're working with a very volatile type of technique. And so this is really a big advancement in what we do in EM. Now, going a step further, when we have all these different orientations in 2D space, we can actually create a three-dimensional reconstruction of the protein itself. And what we get out of that is we get this density map. And so this is the cryo-electron density in three-dimensional space, and this is the actual experimental data that we really get from the experiments. And once we have this, we can build a model of all the different coordinates of all the atoms that are within that protein. And that's at that point, we say we've solved the structure at that point. And then from there, we can make all sorts of analyses and inferences and try to figure out what's going on in the chemistry and the biology of this kind of sample. And this is why the technique is so powerful, is you get this sort of information out of it. Okay, so going away from EM, I'm going to talk about the class of enzymes that my lab works on. So we work on AAA ATPases, and putting it simply, this is an enzyme that exists in many different types of forms in the cell. There's about 250 different species within humans, and they use ATP, so adenosine triphosphate, as a fuel, and it's actually a molecular motor. So it hydrolyzes ATP. It loses that last little phosphate at the end of it, turns into diphosphate. And then by doing that, it releases all its chemical energy, and it allows it to perform mechanical work in the cell. So these things are moving things. And this is a video that just came out from a preprint of another graduate student in the lab that kind of shows about sort of the activity that it's doing. So just really quickly, this is the lawn AAA ATPase, that's what it's called. And this green thing right here is a protein substrate, so it's acting upon it. And then as this thing hydrolyzes ATP, it goes through these different conformational changes or spatial changes in the ordering, allowing it to move around, and then it's grabbing onto that substrate and it's translocating it through the pore. And this substrate doesn't necessarily have to be proteins. This could be nucleic acids. This could be other like molecules of interest, that kind of thing. But that's kind of the basic idea of what these enzymes do, motors. So if you look at the cell, so I told you that there's a whole bunch of different types of these guys within the cell itself, and they're doing various different activities. And this is just a nice graph or a diagram to kind of show like sort of the diversity of stuff that you can do. So these molecules can act upon cytoskeletal elements. These are like microtubules that basically give structure and stability to the cell, and they'll rip apart the microtubule. So it's a severing enzyme, so it breaks one microtubule into two. Another example is this enzyme here, HSP104, which is to the right of the microtubule severing protein. This guy is a disaggregase, so it acts upon misfolded proteins that have really gone awry and have created these like really nasty aggregates. You see this forming a lot in like Alzheimer's and Parkinson's. And what it does is it acts upon these things, rips them out of the aggregate, and allows it to refold to come back to what a healthy protein is, basically. So it's kind of like it cleans up shop. Another example are ATPases that are membrane bound. This one is AFG3L2. This sits in the cell membrane, and it grabs onto membrane proteins that are either marked for degradation or perhaps they're misfolded. It rips it out of the membrane, and it actually proteolyzes or cleaves off and breaks apart the protein itself. This allows for the amino acids to be recycled, and it could be new proteins that are built from it. Likewise, we also have this monster, which is at the very end, the proteasome. So this is the garbage can of the cell. Most proteins, as you go through like the lifetime of the cell, will eventually be degraded and marked for degradation. And this thing just grabs on to marked proteins and just rips them apart. So again, you have this sort of recycling action going on. This time, these aren't membrane bound for the most part. So we're going to talk a little bit about the first category that I mentioned here. So my protein, spastin, that I work on, is a cytoskeletal remodeler. So this is the thing that rips into microtubules. So these are microtubules, these long, like filamentous biopolymers. They're constructed of tiny little proteins called tubulin, and they form these sort of linear, cylindrical structures within the cell. And then you have these enzymes called microtubule severing enzymes, which is this like little donut spider that's right there. And these things act upon the middles of microtubules, and like I said, they actually rip out the tubulin, and it allows for the microtubule to be severed or cut in half. And so they have a lot of different reasons of why we want to do this. I mean, you might think of this as sort of a destructive kind of activity, but destruction also breeds creation in a way, and biology is all about that. So it actually increases the overall mass of the microtubules within the cell. So microtubules grow on the ends, like right here. This is actually a polymerizing microtubule, and it only happens on the ends. So if you cut one microtubule into two, well, now you have two things that can grow instead of one. So you can increase the overall mass of the cell, cytoskeleton from there. Another thing is that this is involved in cell division. So you might remember just from general biology that like during mitosis, for instance, you have chromosomal segregation, mitotic plate. They actually get pulled away. What's doing that? Well, it's the microtubules that are doing that. This enzyme rips apart the microtubules on one end, so it shortens them and allows for the translocation of chromosomes to be separated. It's also involved in wound healing and axonal development. So this is really important for neurons that are really long. They have these super long microtubules. You got to regulate that. Spastin regulates that. Okay, so spastin actually wasn't discovered because of its function. It actually came because of its dysfunction. So this came from a disease. So this sort of family disease is called hereditary spastic paraplegia. It's a neurodegenerative disorder. It affects about one individual in 300,000. So it's not rare, but it's also not necessarily uncommon either. And what it does is that over the lifetime of an individual, that you actually get spastic paralysis in the lower limbs. So lower limbs require these long axons that go from the cortical spinal tract down to the legs. And if you have dysregulation of the microtubule, eventually you can get paralysis by midlife. And it's not lethal, but it definitely lowers quality of life, and it is an unmet need in medicine. So this is an example of like what, I'm not going to show humans here, we're showing mice, it's a little bit more PC, but in a healthy individual, this mouse, we're just going to watch it walk on a plank from the backside, and this is a happy mouse, it can do it just fine. But in the disease model, or a mouse that has HSP, you can see that the spastic gait that comes from this kind of mutation, and also it's not necessarily doing too well in those lower limbs. So this is kind of an example of what HSP looks like. So we really don't know a whole lot about this disease, especially in the form of spastin. And so what's kind of limiting our understanding is also from a structural perspective, is we don't really know what the enzyme looks like in detail. So that's the impetus of my project. So before that, before we came along, there was tiny little bit of information known about the structure of spastin. So you remember I showed you kind of those spider donuts earlier? These things form ring-shaped structures. And so what was solved previously by X-ray crystallography was actually one subunit, or 1/6 of the actual active form of the enzyme. And while this is an important advancement of knowing, like, well, what does this thing look like, this actually isn't the active form of the enzyme itself. You need 6 copies of this to come together, form a ring, and that's what's active, and that's what rips apart microtubules. This thing doesn't do anything. We also have a little bit of a low resolution information that came from small angle X-ray scattering. This is another biophysical method that kind of gave us an idea of what the hexamer looked like. Not a lot of detail, not a lot of information was inferred, not enough to figure out what's going on with the disease. And so that gives us some open questions. So first of all, what does the enzyme look like when it is in the active form? How does it actually engage with the microtubule? Because that's not really known either. And then how is information transmitted through the hydrolysis of that fuel, ATP, transmitted through the enzyme, and how does it basically use that to sever the microtubule? Because we don't really know much about that either. And all this is involved in the disease as well. Okay, so we did EM on it, and this is the result actually. This is the density that came from the hexamer of spastin. And so what I've got here is I've colored all the different subunits in sort of this like rainbow configuration. And another thing that we have in our structure is this green thing right in the middle. That's actually a section of the microtubule that's bound within spastin. So we can start getting some information about how it interacts with the microtubule through this sort of interaction here. The density was of decent resolution, or basically of how well we can tell things apart. It was high enough that we could build an atomic model, which really allows us to do sort of the analyses that we need to be able to really infer a lot of information about this enzyme. And so that's what I'm showing you to the right of here. Now, we knew from some biochemistry that spastin interacts with the microtubule, which is this like half dome, primarily through the C-terminal ends of tubulin. Those are these like hairy little bits that are coming out the end of it. And that spastin grips these C-terminal ends with these things through its pore called pore loops. So these are these like kind of unstructured coils that we know are important mainly for severing. We know that it helps a little bit in actually binding the microtubule, but we know that it can't actually sever the microtubule without these guys. So we knew they were important. And our structure actually has a lot of information about these pore loops and how it's interacting with the microtubule itself. So this is the structure again, the model of spastin. and that I've color-coded all the different pore loops that are surrounding the substrate, or the piece of the microtubule, the polyglutamate chain that's within that. And when we actually look at the density of all these pore loops, we can see that there's a clear structure that's arising from them, and they actually seem to be forming a really interesting organization. So we have this sort of double helix that's occurring between pore loops 1, which is in blue, and pore loop 2, which is in gold, that are sort of wrapping around in this kind of intimate caress of like the substrate itself, which I'm showing in green here. And then the third microtubule, which is in purple, is also forming sort of a spiral staircase that's peripheral to the substrate, but it's not really making a direct interaction there. So it's kind of interesting that it is so well ordered, so there's probably something going on there. And then here's just a top view from that, and you can see kind of the spirally sort of organization, which is no surprise because the whole thing is a spiral itself. Okay, so I'm just going to dive a little bit more detail into the interactions here. So this is the double helix that I was showing you before, and this is color-coded by the different subunits they're at. So I should mention that this is a homohexamer, so every single one of these monomers are identical. It's the same sequence in that case. And so what you see is kind of a repetition of these pore loops that's making the staircase around the substrate, which is in green. And then just going a little bit into the atomic details about these interactions, we can see that first off, pore loop 1, which I've got in blue here, my pointer isn't really working that well over there, so you just have to look at it at this point. So they've got these bulky aromatic residues, which are kind of these tyrosines that are basically creating the sandwich around the acidic side chain of the substrate. So these are these bulky aromatics. They're forming this type of pincer-like structure around every other side chain in that case. These are likely the residues that are responsible for transmitting power. So as this thing is pulling on substrate, it's doing it through these tyrosines. And right in between that tyrosine sandwich, you also have a lysine. So this is a positively charged amine side chain. Let's see if I can get it on. Yeah, it's not going to work. But it's forming this pi-cation interaction in between the tyrosines, and it's also neutralizing the negative charge of that carboxylic acid moiety as well. So it was quite interesting. On the other side, we have pore loop 2, which is the gold structure there. And so these pore loops are actually highly positively charged. All the residues that are involved in there all have a net positive charge, and they're interacting with every other residue. And what we think it's doing is that it's neutralizing the acidic charges on that side, and it's actually allowing it to sort of identify this type of substrate as the microtubule itself. So microtubules are highly negatively charged in that case. You don't want an enzyme that's going to rip apart everything, right? I mean, you want to be able to highly regulate something that could be possibly damaging to the cell. And we think that pore loop 2 is recognizing it specifically that this is a microtubule itself. We think that's what pore loop 2 is doing. Now, going back to that interesting organization that I showed you, that kind of spirally thing where you have also pore loop 3 coming in, we looked back at the difference between our structure, just one monomer, and the crystal structure that I told you about that was solved back in 2008. So their structure didn't have substrate, it had no nucleotide bound, it was just basically naked spastin alone, whereas ours has both substrate and nucleotide. And if we compare our structure with theirs, we see some pretty big differences in the pore loops themselves. So first off, pore loop 2, which is the gold one, is completely disordered in the crystal structure, meaning it's waving around, it's a floppy noodle in space. Emily's lab would probably love to look at it, because that's what they're into. But it's not really ordered, and there's nothing that's really ordering it. Now in our structure, it actually, comes in, it's one conformation, it's doing something. And we also notice that all the other pore loops are also moving as well, meaning that the substrate is influencing the structure to become a new conformation here. And so we looked a little bit closer, we actually found that there's an entire network of residues that are communicating back and forth between these different pore loops. And they're all charged, they're all electrostatic. And if you look at it, it's pointing directly from substrate all the way to the bound nucleotide. So what I've got here in kind of stick depiction is the adenosine triphosphate, ATP. And that last little residue, N629, is pointing directly at the gamma phosphate of that molecule. That's the phosphate that leaves after it hydrolyzes. And so if this goes all the way back from nucleotide all the way back to substrate, we hypothesize that this might be a way for the enzyme to actually sense not just nucleotide state, but also whether substrate is bound, and it might be communicating back and forth between the different subunits. And so we tested it. We tested it through biochemistry. So what I'm showing you on here is an assay in which we have a cover slip, and on that cover slip we have immobilized microtubules they're the ones that are shown in white. There's a fluorophore attached to an antibody that covers the entire thing on it. And every time that you see a blue arrow, that's a severing event that's coming from spastin. And so with the wild-type protein, within about 30 seconds, you have a ton of blue events, right, severing events, and the microtubule is almost completely destroyed by the end of this assay. If we mutate out this arginine 600, which I've circled right here in this depiction of the Pore loop network, to an alanine, which loses its charge, it's way smaller, it should lose the connection between the Pore loops. We can see that even if we have a 5-fold increase in the amount of time that we give this enzyme to work, it's been completely disabled. There's no microtubule severing whatsoever. And if we do an alanine walk to the rest of these residues, we actually see that all of them knock out severing, with the exception of this glutamate 633 here, in which you have a very large decrease of activity. I think that there's probably a water molecule that might be coming in that's actually allowing this interaction to take place, and that's probably why we have decreased activity, but not eliminated activity, but I haven't tested that itself yet. Okay, so what does this mean for disease? So we know, I told you that spastin was discovered because its mutation leads to disease. So we went through the literature, about 30 years worth of human genetics, and we found all instances and all mutations that lead to HSP in humans, and we mapped them to our structure here. And we've actually found 4 different classes, or at least mechanisms, that we think that this kind of destabilizes the enzyme and affects microtubule severing. And what we believe is happening is that any of these HSP mutations are deactivating spastin by preventing it from severing the microtubule. This could be because it can't use its fuel, ATP, anymore, can't bind it or sense it, for instance. It's not being able to use the Pore loop network, which, by the way, is highly enriched in these mutations as well. Or it's preventing it from coming together and forming this sort of active hexamer, which is the active form of the enzyme. And so we hope that our study would kind of be a jumping off study for others who can kind of understand this disease and hopefully lead to a treatment. And that concludes my talk, and there's a couple people I need to thank. First off, I want to thank Gabe Lander, my advisor, and everybody that's involved in the Lander lab. Also my collaborator at the NIH, Antonina Rolmakak, and her lab. I have to thank everyone at the Hazen EM facility, Bill Anderson, Charlie, JC, and Lisa, who are responsible for making sure that the microscope is running, that we have all the computational support that we need, and that we get our data that we want. I also want to say thank you to the organizers of this meeting. Thank you for having me.